Deinococci, or Deinococcus thermus group. This is another one of these small groups without very many species in it. And in terms of genera, this is it. You probably haven't heard of very many of these organisms. Most of these are single species or a couple of species. Even Thermus and Deinococcus, there aren't very many species in them. But they are, these two organisms are fairly well studied. They are deeply related to each other. They're not close relatives at all. And so this major phylogenetic group really does fall into two distinct phenotypes, the Thermus phenotype and the Deinococcus phenotype. And they're quite different. Within each group, they're a lot more homogeneous. So let's talk about Deinococcus and relatives. How many of you have heard of Deinococcus before? Yeah, so most of what you know about Deinococcus has to do with DNA repair. And this organism has been studied mostly because of its really powerful DNA repair systems. The Deinococci are aerobic heterotrophs. They're mostly mesophilic. Original isolates of Deinococcus were isolated from irradiated meat. And so when you can foods, a lot of times they're no longer heat treated because that degrades the quality of the food. But the cans go one at a time past a cesium source and they're blasted to kill any bacteria in there. But they do sometimes spoil all the same. And the most common cause of that spoilage is Deinococcus because it's extremely resistant to radiation. This is a kill curve of gamma radiation comparing E. coli and Deinococcus. And this is a logarithmic scale of survival. And so at doses that obliterate E. coli, Deinococcus has an order of magnitude before it shows any effect at all. Gamma radiation causes double-strand breaks in DNA. As the particle flies through the aqueous environment, it creates radicals that literally break DNA into two pieces. How is this organism able to survive that? Well, first of all, ask the question, is this DNA somehow resistant to that breakage? And the answer is no. If you take cells from this irradiated stuff and break them open gently and look at the DNA, it's in pieces. And yet, they can survive. So what happens? If you take these cells and take a culture of them, blast them like that, and then look at their DNA over time, first of all, the cells stop growing. They stop growing, they stop dividing. And if you look at them over time, those pieces start to get bigger and bigger and bigger until they're full genome size again. And so it's not that they can protect their DNA, it's that they can repair it. And they do this by homologous recombination. So these organisms keep a large number of copies of the genome in the cell and use homologous recombination to reassemble all the pieces. That's pretty amazing. They also have some pigments in them that keep them, that absorb a lot of UV light, for example, and they can repair all kinds of strands. What's the habitat of this organism? It's certainly not gamma irradiated cans of meat, right? You don't evolve an organism for that. In the wild, it's been most commonly isolated from rainwater, gutters, that sort of thing. Why would an organism be so UV resistant if it needs to live in gutters? Its normal habitat is probably water droplets that make up clouds. And so they're constantly blasted by UV light and harder radiation from above. These organisms stain gram-positive, but they do have an outer membrane. And so they are actually gram-negative in the more formal sense. They're non-modal cocci generally. This is showing cells of Deinococci radiodurans going through division, and they have a really interesting, it actually turns out to be much more common than you might think, division process. First of all, notice the genomic DNA. This is a good slice of it right here. It looks like a donut, right? That's real. It forms a toroidal structure. The chromatin is condensed, but these are many, many copies of the genome circle, all kind of condensed down. And this one is waiting now. The division planes are waiting for the thing to segregate so that the division plane can go. Notice that these division planes, it's not actually a pinching of the cell, but you get what amounts to like a camera iris that closes in. So in 3 dimensions, this is like a circle that pinches off like an iris on a camera. These are called division septa, or excuse me, division curtains that divide off. Interestingly, you'll notice, so this is the oldest division plane in this cell, and the cells are actually beginning now to pinch off so that they can separate. The next division occurred at right angles to that. And the current divisions are happening at right angles to that. And so this organism, when it divides, alternates between division planes, right, left, left, right, right, left, left, right. And so this is a kind of a tetrad that's generated in the process. This has some really interesting cell biology implications. Lots of organisms do this, by the way. One of the most important things in animal cell development is an establishment of polarity, right? Dorsal from ventral, anterior from posterior, etc. And there are really important genetic controls for controlling those, so that cells know where they are in that embryo and can develop appropriately. The same thing is happening here. The cells know in some kind of genetic physiological sense their orientation so that the division planes occur in the right way. Thermus. These organisms are typically rod-shaped or filamentous. They're typically thermophiles. They're commonly found in neutral pH hot springs. I showed you some of the habitats for these in Yellowstone, these neutral pH springs where you have pink filaments growing. Thermus is in that zone of the spring. These are all heterotrophs. I would have liked to have shown you some in lab, but thermus isolations aren't working. Yeah.
So it's all the same phylum?
Yes. Yes. So both of these there's 2 branches to this phylum. These are the 2 branches. And again, they're phenotypically quite distinct. Thermus is well known because it's a source of Taq polymerase or TTH polymerase, Thermus thermophilus, which is a little more thermophilic. Interestingly, most of the time when you do PCR these days, you don't use Taq polymerase. How many of you guys have done PCR and know about the polymerases that you have? What polymerases did you use? Do you remember?
Well, when I was doing PCR reaction, we did communication part in that communication studies, and used just Taq.
Yeah. But otherwise?
But otherwise, we used
TFU?
no. I used more.
TTH?
Yeah, we use Taq. There's another one.
So most people are going to archaeal DNA polymerases, and there's a couple of reasons for this. One of them is that it's a dumb reason, right? The patent for PCR and Taq polymerase specifies a molecular weight range, and the archaeal polymerases are bigger than that. And so you avoid the patent litigation issues. But also, these polymerases have proofreading activity, and so they're more accurate and less likely to introduce errors in a PCR reaction. Taq polymerase doesn't have 3' to 5' exo, and so it happily puts in mistakes, and you can use it for that. You can diddle with the salt concentration and tune the error rate in Taq polymerase to be anything you want. And there are times when this is useful. Also, the archaeal DNA polymerases lack reverse transcriptase activity. Type polymerase doesn't have much, but T. thermophilus does. And if you're trying to do a reverse transcriptase PCR, you'd really like to know that you started with a DNA, or excuse me, if you're doing a PCR, you'd like to know whether you're amplifying from DNA or RNA. It's really important. And then there's a bunch of hydrolases and so forth that are used in industry from these organisms. And we already talked about using them from Thermatoga as sources for X-ray diffraction studies of protein structure. These things are typically filamentous in nature, but show up in culture as rod-shaped organisms. A lot happens to organisms when they are domesticated and grown on plates and all that kind of stuff. And most organisms can alter their shape somewhat depending on conditions. And the result is a lot of organisms that are filamentous in the environment are rod-shaped in culture. Yeah? Yeah, they would be. Most of them produce carotenoid pigments, and so you get yellow, orange, or pink colonies. And this is me and octopus praying on a humid day. It looks really dramatic that way. A little bit like Thermatoga, the outer membranes of these organisms are pretty open. There's a lot of space between the inner and outer membrane. And what's going on in there isn't clear. Other members of this group do tend to form blebs and vesicles on the outside. And again, what's the function of this is really, really unclear. But this is this is the way Thermus aquaticus looks in culture. Filaments to long rods. A lot of these organisms can grow by anaerobic respiration using nitrate or occasionally sulfate as a terminal electron acceptor. But Thermus aquaticus can't do that. It can only grow aerobically. Yeah?
So is the sheath, would that be a result of, like, are Thermus and Thermatoga, would they be considered divergent or convergent with that?
That would be probably a case of convergent evolution. And this is not a sheath in the traditional sense. So a sheath is a polysaccharide membrane structure that's on the outside of the cell. And typically, the sheaths are really common in filamentous bacteria, and the organisms move up and down the sheath in the environment. That keeps them in a place, it keeps them anchored to something, but it allows them to move to wherever the resources and things are. Oops.